So this is one of my favorite views of really Dotmatics and Luma. And to me, it speaks to one of the great advantages that Dotmatics have. We have around the outside of Luma all of these best-in-breed apps. What we have here are interfaces across desktop tools and web tools that scientists, many millions of scientists, use today and love. So as we've developed Luma as a platform that ties all of this together, we had a choice to make. Should we rewrite some of these tools as native Luma apps, or should we leave these best-in-breed interfaces alone and just take away the pain of moving the data between them? And I'm happy that we chose the latter. I think if we'd chosen to rewrite many of these apps, it could have taken a long time. And we might not necessarily have got it right anyway. So this is one of my favorite views because these are not just partners. We are all the same company. We can control the software development in these companies, and we can control the interfaces. The demo I'm about to show you crosses a lot of these products, actually. So bear in mind, I'm going to be wearing many, many hats. I'm going through a full life cycle of antibody discovery and some development here. So for you as an end user, you will just live in your favorite app that gets you to your workflow. You won't have to worry about the data flowing anymore. So the workflow I'm going to show you is our antibody workflow here. This slide, don't try and read it. It's a bit busy on purpose. It just demonstrates really the complexity and challenges that we know our customers face that do this sort of discovery. And this is true of many biologics discovery workflows. We know from speaking with customers that many of you are managing these workflows in Excel and in emails and in PowerPoints. And really, that is not a great data strategy, and it's not going to get you to that AI future very quickly either. Likewise, parts of this workflow are great in a traditional ELN, but parts of it, particularly the protein production, don't lend themselves to rigid workflows. You may have a paper on glass style notebook that handles the data input just fine, but the data structure underneath these really flexible data models often in the paper on glass style just isn't going to get you to that AI future either. So what I'm going to show you today is a more simplified view of this. I like these chevron views because they help me break down a complicated workflow. So we're really talking about animal immunization, where we extract some B cells. Those B cells express antibodies that bind with our target antigen for the project. Then we actually have to kind of filter down to good candidate antibodies before we design constructs that are going to express them. Then we move back into the wet lab again. We move into that protein production wet lab where we've got things like expression and purification. And of course, we're always layering characterization and screening data across these workflows. This workflow really bends back to the start of, science is always circular, never linear. So we're going to focus on a couple of different demos here. The first one is going to be our B cell identification. So this is where we've harvested our B cells, they've expressed some antibodies, and we need to sort through these B cells and antibodies to find populations of them. We're going to be using the Dotmatics Notebook and OMIC today, but we could also do the same in the Dotmatics Notebook and FCS Express, or your own notebook and your own flow cytometry tool. Now, these chevrons in and of themselves are really simplifications of much more complicated workflows. What this workflow really encompasses is the flow cytometer. So you're putting your cells or your samples on the flow cytometer. That produces an FCS file, which is swept up into Luma. Then your analysis software has to pick up that FCS file and do the analysis, generate the gates and the numbers. And then the ELN in this case is where we actually made the request for this analysis in the first place. We can also supplement that request with registration and additional assay data. You can connect external systems if you choose, and you can also pull in reports. And Luma's in the middle, always listening to all of this, gathering, analyzing, contextualizing that data so you can make sense of it. Now, you'll hear about some of this pain from Matthew later, but as we've spoken to our customers that do this sort of analysis, the vertical lines here represent, as we've spoken to people, breaks in these workflows or silos of data or manual steps that need to happen. So we have a lot of customers where their ELN is not very well connected to their instrumentation in the lab. Or perhaps they're storing their instrument raw data in file shares and they've got duplication and they can't find things. It's not very fair data principled. Finally, you have to move that data into your analysis tool. Often that's a copy and paste or a drag and drop. It's not very seamless. And we know that flow analysis can just be the first in a whole cascade of analyses that you do on this data. Getting data out into something like Prism, traditionally it's a bit of an arts and crafts project, moving it into Excel, shaping it, and then loading it into Prism. And then finally, just collecting all of the data across all of these silos into something like a dashboard. A dashboard is hard. And all of these silos represent FTE time that is really just being wasted. If you have to reanalyze any of this data, you have to move back all over these silos, find the raw data, and start again. And that's obviously not good enough. It's not scalable, and it's not going to get you to that lower cost and AI future that we're all looking for. So we've replaced a lot of these silos and made it really seamless. And this is going to be the first demo. So we're going to start at the end, really. I always like to do that in my demos. I'll talk to you about a completed workflow where we've got all the data collected. And then we'll step back over all of these various parts, and I'll show you how we generated all of that data as well. So I'm starting off here in my Dotmatics ELN, and I'm going to navigate to a notebook page that has my flow data analysis in it. So the first part of my page is all about setting up the aims and methods, how I'm going to treat the cells that I want to run through my flow analysis. I then have some links out to the actual samples. These come from our registration system. And then underneath here, I've made a request for flow analysis. And this is all encompassed by a single request ID here. That's a really important bit of metadata that we use to tie everything together. The key point here is that the analysis has already been done, and everything south on this page is automatically coming back into the notebook or the dashboard without an email, without any transcription, without digging through file shares. We're breaking down silos even here. So what I have here is tables of metadata that have come from my analysis. And I've got lots of links out to various pieces here. So I can double click here, and this is going to jump me out to our Lab Connect system. And it's going to give me a direct link, a direct download to that raw file. So this is really cool, right? Because from the ELM where I requested this, I can track all the way back to the raw data in Lab Connect. So this is very fair datable. This is findable. This is accessible. It's interoperable, and it's certainly reusable because you can just make use of it from here. Same thing with the actual analysis software. I can double click on an omic link here, and I can jump straight into the analysis workflow from where this data was generated. So I can always see what went on, what decisions were made during the analysis. I have some numbers directly from the gated analysis. And then omic and lots of analysis software can create reports. So I can even link out to those reports directly from my notebook page as well. So here's a PDF of a typical omic report, and I'll show you how this gets generated shortly. Have some additional data around the samples that we used in this analysis. And as I said right at the start, the flow analysis is often just the start, and you may want to do some additional follow-ups in things like Prism. So here we have the Prism graphs coming directly back into the dashboard or the notebook. As I said, we're all one company here, so we are ahead of the game in terms of Prism integration. One of the great things about the new Dotmatics is all the collaboration across all of our companies. So we are ahead of the game. We're still open, but this Prism chart was not exported and imported into my notebook. This was Prism was loaded back into Luma. The data was parsed, and we can regenerate these graphs at that point. And I've got some slides on Prism and where we're going with that later. So how did we get there? How did we get from a simple request for flow analysis to all of this data coming into my dashboard or my notebook? So really that starts in terms of the digital journey. It starts after the flow cytometer has run, and as we're sweeping up data into the cloud, into our Lab Connect system. So this is Lab Connect. This is the web interface to our instrument data capture system. Each of the rows down here you can think of as a connected instrument. And all of these instruments have a Dotmatics watcher attached to them that sweep up files, make those files available in the cloud here and available via the API, so very fair data compliant. You can set things like watch paths, file exclusions. You can even remotely administer all of these watchers. So for IT folk out there, you can update, you can change the way that the watchers detect files. You can administer all of this through a GUI or via an API. So you won't have to log into, your fleet of instruments to manage these agents either. It's really important. So as these files are swept up, as I said, they're made available in the cloud and via the API. We can see here a connected cytometer that has swept up a whole bunch of FCS files that are ready for analysis. Now, even though we could download these files and go and load them into our analysis software, we don't want you to do that. That's an extra step that you shouldn't need to do in a digitally transformed company. So over in Omic now, this is one of our best in breed flow analysis tools. I can create a new data set. And instead of having to dig through a file share, manually find these files, I can use our file actions and I can hook directly back into LabConnect. So now I'm navigating that same hierarchy of instruments and folders that I showed you in our LabConnect system, choosing an instrument, choosing the files, and pulling them directly across into my analysis. Another important point here for fair data principles, and also as you move towards submissions and NDAs for drugs, these FCS files are not being moved. They're stored in LabConnect and Omic is reading from them. So there's only ever one source of truth for these raw data files. And that's really important. So Omic is now reading from them. Once this is all complete, we can move along with our workflow. So the first thing that I want to do is I want to include some metadata in this analysis. I can do this manually. I can do this via an API. I can import directly from the file. So each of these FCS files has been parsed and we have all of the metadata available. Somewhere in this list is my request identifier from my notebook, and that's how I'm tying the two systems together. So I'm going to import all of that in, and now I'm going to move on to a workflow. Workflows, you can build them up as you choose, but we like to use templates. So templates can drive consistency in your organization, or actually if you're using a CRO to do the primary analysis here, you can give them a lockdown template so that you can compare apples to apples because we're all using the same workflow. So we've already saved a lot of time here, a lot of man hours and effort finding files, making them available up in the cloud, organizing them, getting them out into our analysis software. But as Kaleem mentioned before, we've got lots of other techniques while you're in a scientific software like this to speed things up and drive consistency. So in this case, I have that auto gating enabled. So I can click on my auto gating system here or my gating. And instead of manually drawing polygons around these cell populations, that's the kind of art, if you will, of flow data analysis, often done by humans, often takes time. Humans can be inconsistent. We can just do this AI or machine learning auto gating that will either train on your best gated data set or in our next release, use just general machine learning to draw gates. And this is going to speed up a lot of time and drive consistency. If I don't agree with the computer, I can move the gate around and move along with my day. There's lots of other detail in here, obviously, and call for a demo of Omic if you want to get more detail on this. And then finally, we've got a couple of figures and exports in here. So the figures basically sets up the PowerPoint, the PDF that we saw delivered back into the notebook. And the export sets up the metadata and the numbers from the gating analysis to be pushed back into the system. Also from the exports tab, and I'll move across this quite quickly, we have another time-saving piece in here. So the time-saving piece is, as I said, this analysis quite often is just the primary analysis and there's often follow-ups. So I can export a CSV and that's what I used for this workflow, but I can export directly into Prism from here. So Prism, best in breed charting and analysis tool. but often you have to do some data manipulation, often in Excel, to get the data into Prism. We take all of that pain away here. We write a native Prism file, and we can send all of the data across. That looks a bit like this. So we send it across into a table. Then you can set your own analysis up. And as I'll show you a bit later in this, we're also working on send to Luma buttons from Prism, where the file will be sent across, the data will be passed from that file, and we will regenerate these graphs that you can use in the dashboards and things that I showed you before. So once I've set all of that up, I can go back to my workflow, and with a click of a button, I can send all of this data off into Luma. So when I do that, each of these data sets gets a unique ID in Omics. So this is a unique data set ID. What we do is we send these files across. So you don't have to download the files and upload them into LabConnect again. You just go over into Luma LabConnect, and that has already been sent across. So we have a folder waiting for our Omic results. Each of the subfolders is one of those workflow IDs. And we have a number of files in here now. So we have that PDF that I talked about before. So this is the charts and the graphs of the gates that we've done in our analysis. And we have the CSV of the results and the metadata. Now, as Thomas and Clem have both mentioned, as you move a file through these digital transformations, the value of that file increases. And LabConnect plays a really big part in that journey as well. Not only does it make files more findable and accessible, sweep them up, make them available in the cloud, it also makes them more interoperable. And it does that by parsing these files. So if I go and look at the parse data here, we have parsers for kind of basic file types like CSVs, as I'm showing you here. But we also have scientific parsers for mass specs, NMRs, flow cytometers. You name the instrument type, we've probably got a parser for it, or we'd love to work with you to create one. For data scientists in the room, we convert these files into JSON representations of the files, and that makes them more transportable and usable across different softwares. But for the bench scientists in the room, this is probably an alien format, and you get to a so what moment quite quickly. So what, you've got this in a JSON object. I want it in a way that I can read and understand and put it into dashboards. And that's where Luma comes into play. So it integrate, Luma and LabConnect are very tightly integrated. They're really the same software. And Luma can take these JSON representations and add more value to these files. So over in our Luma interface, what we can do is we can build data models that we want to flow this data into. So think of each of these boxes as a table in our data model. It's a table in Databricks, our chosen partner for all of this work. And I've built this rather simple data model to kind of mimic how the data comes out of Omic, so I can use it in dashboards. So I have data sets. That's really the folder of data. Within a data set, we generate files like the PDF. We generate results from the gating, and we generate metadata. Now, if you were eagle-eyed, you'll have noticed in those Omic folders in LabConnect, the raw FCS files were not in there. Again, we don't move them. There's only one copy. But in the metadata, there's a link out to them, and that's how we're able to draw this data model together. But we've still got JSON representations of these files, and then this nice data model. How do we get the JSON files and break them apart and put them in here? The secret sauce in Luma is these raw data flows. These flows can be SQL-based, they can be Python-based, and in the future, they will be just click and drop, really. So you'll be able to find a part of this JSON representation of the file, click on it, and map it directly to one of those tables in the data model. So as the analysis happens, the analyst clicks on the button, they're done with their day, the data moves into LabConnect, flows into Luma, into this data model. And then, as Kaleem said, this data model is open, so I can get at this data via GraphQL, REST API, or JDBC kind of direct database connections. And that's how we get this data back out into the ELN or the dashboard, as I said before. Okay, so I'm going to move on to the next part of the demonstration, but if any of this resonates with you, if you're a company that's struggling to connect their notebook and registration, and instrument data in a fair data principle compliant way. Put your hand up. The marketing department is in overgear here. We have an email address called luma@dotmatics.com. Email us. We want to help you with this. Let's build this kind of data forward platform that you need to get to the AI future today. So we'd really love to speak to you about these things. Okay, so let's look forward in Luma. So some of this is going to be roadmap, but it's pretty near term. So we've got the software and we're happy to show you it. So far, what we've been talking about is kind of how we're interoperating all of this software to move the data into Luma and start to make sense of it. We've really just been talking about a small slice of Luma. So we've been talking about data management and processing, really foundational, absolutely vital to getting to you where you need to go, and data and instrument integration on top of that. But I want to show you some of the other pieces that we're working on, and I'll start with AI. I already showed you some AI in the AutoGate in OMIC. That was some of our scientific domain-specific AI. And I want to step out in that differentiation ring that Kalim talked about and show you some AI actually in Luma. We'll build on the flow cytometry example, but this time we'll imagine we've got a whole fleet of cytometers generating lots and lots of data, and I need to work out how much data I've got and where it is. So I'm just going to jump back into Luma and I'm going to go to a slightly different app here. Now, this app is built directly on the FCS file standard. It's not built on the simple results flow that I showed you before. And you can see it's already a bit more complicated than before. Now, if you think about data models for things like mass spec or NMR, these get even more complicated. And as soon as we build a parser for a given instrument, we build this data model automatically. So that's convenient, but this model is a bit harder to find data in. So how can we make that better? So what we did was we trained a well-known large language model on this data, and we have enabled kind of AI searching on here. So I'm asking a question in here. So find me all the cell markers, that's what CD is shorthand for, across all of my instruments, and group by the number of files and show me the largest first. So it's looked across my data model, it's written the SQL for me, and I can go and run this SQL directly. So this is like a huge time saving. This is just a demonstration though. Imagine when we're showing you the dashboards that you could ask a question of a really rich dashboard and it just filters down for you to the data of interest. Imagine not having to dig over different data sources and write specific queries, just talk to the computer and it gives you the result. So it's a POC right now, but it's really exciting that we can do this sort of stuff on top of these complicated data models. Okay, so next I want to talk to you about material management, adaptive workflows, and as Kalim mentioned, that's a really purposeful word, adaptive, and tailored experiences or dashboards. So if we move back to the broader workflow, we're still only 2 chevrons in, and we've got a lot of ground to cover here. So we've already found our B cells, we've extracted them for our animals. We know the antibodies that they express. We've run those expressed antibodies through a sequencer, something like in Illumina, and Lab Connect has swept up all of that sequencing data. So now we're moving into the part of the workflow where we need to first filter through all of those candidate antibodies for ones that we think are going to be good drugs. And then we need to do some design, some molecular biology to generate constructs that will express those. So let's go and have a look. I'm moving into Geneious now, which is part of our family of products. It's Geneious Biologics here is a web-based tool, and it's designed primarily around the antibody workflows. So what I'm showing you here is sequencing data that Lab Connect has swept up and made available. And I've got read quality graphs behind my sequences here. Remember, what I'm trying to do is filter down to good potential candidate antibodies. So my first course filter might be, I want to remove, or I only want to use sequences that have good read quality, that I can have confidence in the sequencing. We have a mixture of heavy chains and light chains, so I pair them next, and I'm going to jump down a step and talk about annotation. So we do annotation for two reasons here. We annotate against either public databases, your own internal databases, blends of the two, for things like regions of interest, like CDRs, the binding regions in these antibodies. But perhaps more importantly, we also annotate against liabilities. So are there any patterns or sequences in here that might not make these favorable drug targets? They either make them unstable or they fold or they're just not good. So once we've found all of that data, we've got our sequences of interest, we can now get this data out to our wet lab. Now, traditionally, as I've said, we know our customers right now are exporting these files, doing manipulation in Excel, maybe into PowerPoint, sending emails around, and really struggling with this workflow. So we can actually add value directly here by just registering this back in Luma. So with the click of a button, I'm now done with my work, and I can go into Luma, and let's go to another app. So this is an antibody app that we are going to use to kind of track all of this data going forwards. And I'm going to show you a preview of our design tools here. So this is going to be a preview of some of the interfaces that we're building in Luma. And all of the data that comes into Luma, we can collect and analyze in here. So the first tab is our registered antibodies. So these are the antibodies that I just finished in my molecular biology tool and sent across into Luma. Notice that we're using the same sequence viewer here. We're going to be using this Geneious sequence viewer across a lot of our applications. It's going into browser in 7.1. It's going into our notebook. It's obviously going into Luma. So we're trying to drive some consistency here as well if we can. Now, one thing that happens is that as I clicked on register there, you notice I didn't fill out any additional fields, and that's because we do a passive registration as the data comes in. I like that actually because this internal identifier that gets generated, all of the downstream data from this ID gets from this sequence gets attached to the internal identifier. And that means that if I do any curation downstream, and chemists in the room, this happens a lot, right? You elucidate the crystal structure of a compound and it changes batch. You won't need to then also curate the assay data because we attach it all to this internal ID. The corporate ID is almost an alias. So it's really going to save a lot of time and headache as you add data to these entities and then have to do curation. So we have our antibodies in the middle here. These are our candidates. Now we need, if we go back into our workflow here, now we need to design some constructs that will express these antibodies so we can move back into the wet lab. So to do that, I'm going to move into Geneious Prime. Now, this is a desktop tool, but just because it's a desktop tool, it doesn't mean that you can't get the same data flows and experience that we have in some of our web-based tools. So in this workflow, the antibody picks are the antibodies that I just generated in Luma. And then I have my vector here. Now in Geneious, there's lots of workflows in here. I talked to you about workflows in Omic and how they can improve and kind of help speed things up. We have them in here as well. Geneious is its own demo in and of itself. I'm going to do some parts cloning today to generate the instructions really for the constructs that will express my target antibodies. Now, the way I like to think of this is in the chemistry world, chemists have always had a sketcher at the top of their notebook page and they sketch in their reaction and the data gets parsed into tables for them. Biologists have been kind of starved of that enterprise experience for a long time. But it turns out what we've actually got here is the equivalent to a chemical sketch. We have all of the ingredients and entities required to generate a construct that is going to express my antibody. Traditionally, people will be exporting these files and then going through that pain of managing them in Excel, sending emails around. We can take away that pain with a Send to Luma button. That's going to send the data across back into Luma. And now my wet lab team can pick that up. So back in Luma here, my construct design sets are also being sent across to these dashboards. And I can go and filter down and look at the constructs that are ready for moving into the wet lab. Okay, so we've kind of finished our very quick design session. Now we're going to move into the wet lab. We're going into make actually action on these construct designs that we've just done. So to do that, we're going to move into our adaptable workflow piece. And this is the part of biologics workflows that needs that adaptability. It doesn't need a rigid system. You need to be able to move around different paths to get to the same product, navigate around errors or mistakes or things that crop up. So the workflow here must be adaptable. Otherwise, you're endlessly creating template after template, and that's just not efficient. So let's go to our adaptable workflow. Now, the key currency of this adaptable workflow is tasks, and I'll show you a few of the tasks that we've generated today. Two key things about these tasks. One is that they're scientifically aware, so they understand the inputs and the outputs required. So for example, this mass spec task knows to reach into Lab Connect and pull out parsed mass spec data. Same with this UV task. We're working on tasks that will launch things like Geneious, that will launch things like Prism. So it's going to be a workflow piece, not just for the wet lab, but also for analysis. So that's key point one. Intelligent and scientifically aware. The second point is that they're going to be designed by you guys in a low-code way. So you won't have to write an SOW with Dotmatics or a consultancy firm to generate your own palette of tasks that match your workflows. And you'll be able to add in the right level of adaptability for your processes. So that's really, really important. We're going to work on a couple of tasks today. Let's go and start an order inserts task. We will call it demo. So you'll notice in these tasks, and this is fairly consistent, we have high-level metadata, including things like statuses and assignees. This is data that will drive a Kanban board back in the dashboard. Then we have two different tasks, order and receive. You can name them differently, but essentially it's plan, and then it's get the actual results. The plan in this case is fairly prescriptive, and I'll show you in the next one how we can get much more adaptable. But I'm saying my start point here is one of those construct design sets from my Geneious product. I'm going to pull that in, put in an amount per well. My fill direction doesn't really matter. So for my inserts, I don't care where they land in these plates, but I really care about matching them up with the rest of the construct in the subsequent steps. So now I had, I was putting this in a 96 well plate. I had 106 inserts, so I've got two plates. Each well has a single entity in it, and I can drill down into that well and see my requested amounts from here. Now, there's imports and exports all over this workflow piece. I'm doing these exports manually via files, but just know that you'll be able to configure API endpoints here as well to make this all seamless. So we'll pretend that I've exported my request for my CRO. I'm now waiting for them to do the synthesis for me in this workflow. They've been amazingly efficient, and they've already sent me the plate back, so I can move to my receive tab pretty quickly. So I'm going to import the manifest of data that my CRO sent back to me. And if I just navigate to my file, so that's going to be my inserts file here. In this file, I have the original internal IDs from my inserts. The CRO has generated a plate barcode for me and a location, and I can just load all of this in. And it's going to do two things. It's giving me a real plate barcode now that I can use downstream. And for each insert, it's giving me an actual amount that was generated for me. So I can say, okay, I'm done with this part of my workflow, hit save. This is now driving our Kanban board back in Luma here. So these Kanban boards can reflect what's happening in the adaptive workflow, or actually kick off workflows by dragging and dropping these cards around. So just one more piece in the adaptive workflows, because I really want to show off some of the scientific intelligence in here. So if I go back to my tasks, and we'll do clone constructs, same deal as before, high level metadata on the first tab, and then inputs and outputs effectively on the next two tabs. So my first input, this one is much more flexible, because instead of prescribing it has to be a construct design set, I've said you can pull up a previous task, or if some of the plates that were delivered back had contamination or just weren't fit for purpose, I can pull individual plates into here. So I've now got flexibility into how I start this specific task. I'm just going to pull from the same design set for speed, put my cells in. The real magic here is though that I'm inheriting the fill direction. So I'm going to do this on top of the inserts barcode from my CRO, but I'm going to use the insert ID, match it up with the construct design set and pull in the other ingredients, the other entities that I need to generate this construct directly into the plate and match it with that original insert. So going back to that molecular biology design, now I have my insert from the previous plate. We've done this on the same barcode. And I have backbones and cell lines and water and all of the other ingredients imported directly from that original design. Now, the export and import in here might be to something like a liquid handler, because we know you guys have probably got your cell lines in-house and already plated up. So you could be doing API calls or files to your liquid handlers. Back in Luma, obviously, this has all been driven by the dashboard. Okay, so we know there are a lot more steps in here. I didn't want to keep showing you different task types because the concept is the same. Info tab, essentially, and then 2 tabs of work. But you'll be able to build these up. Luma is in the middle, always listening and collecting all of the data from these. And now, you want to do some screening and optimization and data exploration. So what does that look like in Luma? Let's go back over to Luma and I'll show you our project assay data dashboard. Just an example here. So Colleen mentioned that to do something like an antibody workflow, there are many, many different data sets. And what we've done here is pull in a sample of some of the important ones that we've heard about. So we have the titer concentration versus the ELISA. This chart is actually sized by that liability score that we generated right at the start in our molecular biology tool. I also then have like a ton of Prism graphs that again have been not exported and imported manually. I've just dropped into Luma. We've parsed the data and we've generated the graphs programmatically. And then finally, we've got sequencing data. So this is, ripe for this AI. This is the kind of dashboard that I was talking about before, where instead of having to write a query in each of these data sources to find my data, wouldn't it be cool if I could just type in a natural language query and have the data filter to my antibodies of interest that I need to move on to? Okay, so I've talked a lot, and I'm going to talk to you about Prism since I've mentioned it a lot. But if this resonates with you guys, if you are in a wet lab, we think particularly in these biologics workflows, and you're struggling managing all of this data, you're struggling coordinating handoffs between your teams, you've got data silos in Excel and email and PowerPoint and PDF. Again, come and talk to us, luma@dotmatics.com. We'd love to get your input on this and partner with you to help remove all of these silos that we know you're struggling through right now. Okay, so back to the slides. I've spoken a lot about Prism and our plans for Prism, so let's just dive a little bit deeper on where we're going here. And this is really, a long time in the making. Prism is used by millions of people globally. As I said at the start, getting data into Prism is traditionally a bit of an arts and crafts project. You're in Excel, you're manipulating data before you load it. Also, up until Prism 10, the format that the data was stored in was kind of locked away in this binary format. It wasn't very accessible or interoperable. You'd have to export it. But with version 10, we have a new file format. The data is all stored in JSON. That's the same JSON that I showed you being parsed in Lab Connect. So the data from these tables is now way more accessible and interoperable. But what we don't store in the Prism format is the actual graphs, because the files just get too big and unwieldy if we do that. So as these Prism files land in Luma, we have command line tools, since we're all one company and we're collaborating together, command line tools that will basically regenerate all of these graphs so that you can have them available in dashboards. The other piece of the Prism pie here is that in our adaptive workflow piece, We're going to add some extensions, as I said, and task types that allow us to launch Prism. If you think about the demonstration I just showed you, what we're actually generating here are plate maps. So it's known entities in known locations in wells. It's concentrations of those. We already have Lab Connects, which you can attach to your Envision or any of your screening detectors and pull that across. So what we're planning on is a normalization step where we'll layer that raw data over that plate, do your percentage inhibition, for example, and again, at a push of a button, then package up all of that data into a .prism file. We can associate it with a template as well at that point to drive more consistency in the analysis. And then, as I've mentioned several times, push the data back into Luma, parse the data, generate the graphs, make them available in dashboards. Remember, the ELN dashboard right at the start had Prism data delivered back into it. That's all done with Luma in the center. So I've had to go through this quite quickly. This is, many groups collaborating over many months to get to, an expressed antibody. But just to recap, we talked about animal immunization. I didn't show you our notebook, but we have nice templates for that. I showed you flow cytometry analysis of those B cells. I showed you some molecular biology, some kind of preening of the antibodies, and then design of constructs. We moved into the wet lab with these really adaptive workflow pieces. And then we talked about how you can get assay data, and I showed you dashboards. So again, if this resonates with you, luma@dotmatics.com, get in touch with us, and thank you for your attention.